Hi everyone. Sorry we're late. We're having some technical difficulties today. And let's see. We had to switch over to using the iPad. So bear with us as it cuts our head off, huh? I'm just going to try to get Dr. Dominic connected. Hey Lauren, Nicole, how are you guys? I'm so sorry we're late. I am going to figure this out. I've not done a dual live before, so I'm trying to see how to add him. So just bear with us for just a minute. Hey Jana, how are you? I am messaging with his team so we can get them online. So we're going to share some of your questions. Hey Jennifer, how are you? With Dom and get you guys some answers on the, Ricky, you're gonna help mommy watch for questions, okay?
Can I put my feet over? Oh,
you can put your feet over here. Okay, so I'm sorry if the angle's kind of weird. Hey Dana, how are you? Okay, Dominic, I see you. I just don't know how to connect the two of us. Ricky's gonna try to keep an eye out for questions being answered here. Okay, let me see if I push this, add. Oh, hey. Okay, be on track here. Hey, Mary Jo, how are you? I miss you guys. Okay, Dom, I sent you a add guest. This is super cool. I was trying to get this set up earlier and then everything went haywire. So it says it's adding Dom, and I know he's really limited on time. So I'm going to ask the questions that were submitted. And then if we don't have time for questions that come in, hey.
Hey.
I'm sorry.
I'm so sorry for the technical. So I was just briefly letting everybody know that we were going to connect with you, and I know you're limited on time, so we'll ask the questions that were submitted, and then if I'm sure several questions will come in afterwards, and maybe we can get something up on the website later on, and you can always go back and view and see what people had asked. So thank you so much for doing this for the community. I know appreciate that. So, Dr. Dominic, I see you. Is that Amy?
No, I'm Sarah.
Oh, Sarah's with you. I'm sorry. Okay, so I don't know if you want to do a quick intro and then we'll get right into the questions because I want to be able to get as many of them answered before you have to go.
Yeah, absolutely. Well, I'm Dominic D'Agostino. We're at the University of South Florida, Morsani College of Medicine. I'm an associate professor here, and we study a range of products, a range of disorders, I would say, associated with targeting with nutritional ketosis, and that can vary by, by method. It could be the ketogenic diet, which we've done studies on that. And we also develop and test ketone supplements. And our research was really sort of spearheaded by the Office of Navy Research, which continues to fund our lab to develop things for the warfighter and military applications. But one of the emerging applications is Kabuki syndrome, and we are very excited to be on this project, and it has become the dissertation project, the PhD dissertation project of Sarah Moss here. So I'll let her introduce herself.
I'm Sarah Moss. I started volunteering here in my undergrad about 2 years ago. Kabuki syndrome was very random, and I'm very happy it showed up in our lab because, yeah, I've been very involved. It's been awesome. And I'm very into genetics and epigenetic modulation and research. And yeah.
Thank you both. We're really excited to have you guys doing that work on your side that could potentially help our community too. So if it's okay, let's go ahead and just dive right in. I'll ask the questions as they came in, and then if you need me to repeat or anything, just let me know. But so the first question is, are there any preliminary results from the pilot study? And if so, what are they? Or is there anything that you can share about that?
So the pilot study was mainly for us to to make sure that our protocol was going to work, and to finalize our decision. So we made a lot of progress in that, and we're very confident in our protocol that we have set up now for the main large goals, because we don't want to just start a large project like our 3-month study without trying out the protocol first, I guess, to make sure that the mice will eat the food, the BHB will actually absorb into their system, as we've seen previously, and that their living situation is compatible with them actually exercising and being active. So it's
palatable. So we discovered that the palatability of the supplements are such that they will eat the food and it will elevate the beta hydroxybutyrate levels independent of following the ketogenic diet, just through supplementation, elevate it to levels that we think will be efficacious for this, this disorder.
Okay, awesome, thank you. And Ricky's gonna kind of watch to see if any questions come in related to what we're talking about, and if we miss them, I apologize ahead of time, everyone, but we'll make sure that we get any answers if we miss them. So the second question was, so that was regarding the pilot. The second question is, has the phase 1 study been completed, and do you have any results?
So it has not been completed. We have 3 weeks left, and then we have the end of life analysis, which is when the behavior tests will be run and the tissues will be collected. And then after that, we are going to run another version of this 3-month study starting near January. And then we'll go into the molecular analysis of the tissues that we collect.
So the project is kind of staggered based upon the breeding of the mice. Okay. So we're doing the sample size for the phase 1 of phase 1 would be.
So we were hoping for 32 mice, so that we can have 4 in each group. So we have male versus female, wild type versus Kabuki, and treated versus non-treated. However, the breeders didn't really agree with us on that, so we have to stagger it out. Yeah.
Kind of like our children. Okay, so that kind of answers the third question as far as what kind of data or when the final results for each of the studies might be available. So we understand that you're working on that. Is there any specific data that you hope or expect to collect through this process in the different phases?
Yeah, so our behavioral tests are open field and elevated plus. So those are going to be measuring their exploratory behaviors. Do they prefer to, do the mice prefer to stay towards shady areas where they can hide, or are they more open to going out in more risky parts of the box, basically? And then the elevated plus is the same thing, but it's a cross maze and it's elevated, so they have a risk of falling if they go in the open areas. So it's anxiety and exploratory behavior is mainly what we are focusing on for the behavior. And then our molecular stuff, we're going to be mainly focusing in the brain to do the neurodevelopment and cognition capabilities. And then we're gonna kind of go into other tissues just to see if there's any main difference between the phenotypes of the wild type and the Kabuki that we can, establish.
Excellent. Awesome. Well, thank you. Okay, moving on. Next question. Can you describe how, so you did briefly mention something earlier, but the question is, can you describe how the projects were funded and how the money was used?
Okay, so the foundation, we got a generous donation from the Kabuki Foundation, and that really spearheaded the initial being able to get the mice, house the mice, and keep the mice in our vivarium to support our breeding protocol for, many of the mice we have now. We got over 100 mice, I believe. And USF has a foundation, sort of, the account is linked to my name, and that foundation is also supporting things to match over the course of the project. Will match the funding from the Kabuki Foundation. And we also got a generous contract through disruptive ketogenic supplements. We're also sponsoring a clinical trial on Angelman syndrome, which we think will be a good precedent potentially for doing a clinical trial for Kabuki syndrome. And the funds from disruptive nutrition, not to get too intertwined, actually were matched by the state of Florida too. So we have several pots of money coming in to support the project. Some from USF, of course. The Kabuki Foundation generously donated and got this project up and running. So, and we look at this as a long-term partnership with Disruptive Nutrition to continually support the science and potential application of ketone supplementation for Kabuki syndrome. So we're hoping that, the preclinical studies we're doing now can transition into ultimately a clinical trial. That's the hope for what we have now.
Okay, thank you. We lost you a little bit in the middle, but if anybody has any questions or wasn't able to make that out, just let us know, and we'll go back and get that verified with you. We have a question that's saying, how long of a time period do you put towards behavior analysis, and how do you choose how long you set that time period for?
So we based our behavior analysis off of the literature. So and we consulted a few of the actual behavior experts here at USF. And we determined that those 2 tests we are doing will be the most reliable for the data we are trying to collect. And so we put them in their behavior room for about 30 to 45 minutes to let them acclimate, so we don't have any anxiety from not being in their general environment. And we run them through one test and give them at least an hour break between tests. So we're also doing grip strength. I forgot to mention that earlier. So we are doing grip strength one day just by itself, because we don't do more than 2 behavior tests in one day to avoid stressing them out and affecting the results. So we do grip strength on one day, and then the next day we do open field and elevated plus, and we let the mouse at least have an hour in between the 2 tests.
I think you're
good. Are we good?
They're in a hospital. I think we got you there. So just so everyone knows, they are at USF, and you know how wonderful the cell service is in hospitals. So bear with us on this live. So thank you. I think we got most of that. It froze for just a moment. There's another question here coming in. I'm going to jump back over to the list though real quickly. And you had mentioned that the mice aren't always cooperative, with the reproductive process you guys were going through, and that leads into this question. And can you describe some of the challenges that you've experienced in getting this project off the ground? Oh no.
Can you hear me?
We got you.
Are we frozen? Oh, okay. So there's quite a lot of paperwork associated with this, from the Animal Care and Use Committee to the Material Transfer Agreement to get the breeder mice from Johns Hopkins, to actually sort of developing, the actual protocol. And then, Sarah has done a lot of the heavy lifting on sort of writing things up and logistically being able to sort of pull this off. So do you want to, and she has a lot of experimental challenges that were.
Yeah. They're the mice every now and then, they're mice, right? So they're animals, they run off of their base instincts, engage me because they feel like it. And that kind of throws things off. But so far we haven't had too many of those, and it's been running relatively smoothly now that we've gotten past all of the paperwork parts. And the pilot, trying to figure out what will make them cooperate the most. So it's gotten better, yeah, so far.
And they're breeding.
And they are breeding, yes.
Pretty
so,
steady. We have a good
breeding
rhythm going, I guess you'd say, right?
Yeah, we have a good rhythm. So mice have a tendency with their first litter, they will eat all of their newborns, which really throws a chink in the breeding part.
But that's not uncommon.
Yeah, it's just right. It's what they do.
I've heard that before. That's interesting. One of the questions that came in is, when do you think a clinical trial on Kabuki children could occur in the next 1 to 2 years, more than 2, more than 5? If you had to speculate based on your progress right now.
It will really depend upon, getting the necessary funding to do that. So we really hope to partner with our industry partner, I guess you would say, that's really helping to move things forward in the field of Angelman syndrome clinical trial experimentation. So we have hopes that within the next 1 to 2 years, things can go in the direction of a clinical trial. And we think that the things that are going on now will help sort of pave the path and maybe set a precedent for that. And as we move forward on other projects too, we're learning, ketone supplement. Am I frozen again? Okay.
There, you're back.
Okay. Just to summarize, it's hard to say, but we're moving in that direction, and I think within 2 years, it's possible.
Okay, and viewer is wondering if you can share who your industry partner is.
Disruptive Nutrition, but they go under different names. But Disruptive Nutrition has really been, very focused on the clinical application of ketone supplementation, and really focused on, neurological diseases, and this idea that ketones are neuroprotective and that they can be therapeutic for this disorder and related.
So that viewer said that she works for a big pharma company and she'd love to run that up the chain. So we'll make sure you guys connect.
Yep.
If that's
great.
Okay.
Thank you.
Awesome. Thanks, Jenna. Well, so how will future projects be funded? Other than what you have shared about your partners, do you have any other?
Well, we have our foundation account that has supported projects, and I think it's possible that we could write a grant to other foundations and even the federal government if a funding opportunity becomes available. So because Kabuki syndrome is relatively rare, there's not a whole lot of organizations that will invest in funding these types of studies. So we have to be a little resourceful and creative in how we fund the lab for this kind of project. But we think it has major implications for other potential disorders too. So that's kind of how we pitch it.
Sure. You had mentioned Angelman earlier, and one of the questions was, is there any data yet on human trials of exogenous, I don't know if I said that right, but ketones in Angelman syndrome patients?
Yeah, it is. Well, it's a blinded trial and it's being conducted at a Vanderbilt, and I believe you can find it on clinicaltrials.gov. But the trial is in process now, and they're continuing to recruit subjects into the trial. All I know is that things have been going very well as far as recruiting and as far as, being able to implement the dietary ketone supplement into the diet and into the lifestyle. These things are much, much easier to implement than the ketogenic diet itself. And I think that's really where the innovation and the implementation and ease of compliance is really a benefit when it comes to these supplements.
Okay.
And as far as I know, they taste really good too. So it'll be something that will be enjoyable, potentially, and pleasurable to drink instead of, formulating your whole diet to a different eating regimen, which is, I think, important for quality of life.
Yeah, and I think especially for the kids that maybe they can't go through that process. One viewer is asking, is ketone supplementation during normal diet equivalent effective as ketogenic diet when looking at the cell metabolism?
Well, we think that the ketogenic diet, many of the benefits are through the elevation of ketones, and specifically that is the case with Kabuki syndrome. That's kind of our hypothesis. But we also believe that the ketogenic diet or nutritional ketosis has benefits in and of itself, and adding ketones to the ketogenic diet may have an additive effect.
Yeah, so it would depend on the circumstance as to why you're trying to use BHB. So like if you have a glycemic control issue, then probably not going to be as effective as going pure keto. But if you just need the BHB in your system, there is evidence to support that it might be, but we'd have to run a study to say for sure.
Sure. Thank you. Just a few more questions. One of the moms says that Lorenzo is 5. He's type 2 Kabuki, severely affected. She says his brain, ears, eyes, lungs, heart. It's all affected. He's very happy, and she learns from him every day. But she's wondering if you can talk a little about KDM6A research. Is there anything out there that you're aware of, or do you know of anything coming even in your lab in the future to look at that mutation as well?
Where there's a lot, if any, research directly on type 2. Most of the theories that have come out about type 2 are just relating the mechanisms of type 1 and the results that we have had with type 1 to how it could affect type 2. So theoretically, this could benefit type 2, but we do not know for sure because we don't have a mouse model with type 2. It's going to be very difficult to start the type 2 research specifically. Yeah, so unfortunately, there's not a lot.
But that can change in the future, so
the research tools are not yet available.
For us to be able to actually analyze type 2. Because we got our mouse model from Bjorn. His work is what started all of this. But so far, I haven't seen a mouse model for type 2 out. So.
But, we have studied ALS in the past, and we study the familial form of ALS, which is maybe only about 10% of the population. But the implications for the Diana protocol is what we studied for ALS. The implications are that it would also be therapeutic for those with sporadic ALS, where the etiology is largely unknown, but the symptoms and the the end result are very similar as far as the progression of the disease. So we do think that even though we're not studying from a molecular level that the type 2, we think the benefits that we see in type 1 with this experiment and potentially with a clinical trial, it would not exclude type 2. So I think from a clinical trial perspective, it may be equally important to study type 2, if that's sort of what they're getting at. Yeah.
And Angela from your team has posted a couple of links from NIH just so for you viewers that are on that you might want to check out. We have a question asking what the protocol in Angelman clinical trial, or sorry, what is the protocol in the Angelman clinical trial? Do they not eat anything besides the ketone beverage? Are there other lifestyle changes they need to make besides drinking the beverage? And how many times a day do you think, or do they drink it? So that was a question I just blurted out, but
yeah, that's a good question. So the patients that are enrolled into the trial, they do not vary what they were doing. So they're basically, everything is kept the same. If they're on the ketogenic diet when they enter the trial, they stay on the ketogenic diet. If they're on an anti-seizure drug, they stay on that drug. If they're on its diet and add ketone supplement on top of that. If you can hear me.
We lost you just about halfway through that.
Okay, I can hear you. So the ketone supplements are given several times throughout the day based on the weight of the subject.
Okay, but they don't change anything.
They don't change anything, no.
While they're on. Yep. Okay,
and some of the patients may already be on a ketogenic diet, and if that's the case, they would not take them off the ketogenic diet. Existing dietary protocol or even drug regimen.
The last part of Dana's question was, how long does the trial last? Do you know?
It's on clinicaltrials.gov, so you can look up Angelman syndrome and just type in. So I believe it's 16 weeks.
Okay. And Angela did just post the link to that on there.
Okay.
So.
Okay, great.
Well, thank you, Angela. Thank you for answering that. I know we just have a couple more minutes left with you. Can you tell us? Okay, so one of the mamas in Canada that helped us with our fundraising for your lab says that she would like to know how confident you are in the research. I'm sorry. How confident is the research team that ketones can have a positive effect on kids?
Yeah, well, I mean, we've studied many different disorders and looking at nutritional or supplemental interventions. And one thing that we feel is remarkable about the ketogenic diet is that it is sort of generally accepted that it has neuroprotective properties, and those neuroprotective properties are likely a result of two things. One is that the ketone bodies themselves are serving as an alternative fuel for the brain, and they really do impact brain energy metabolism and the overall, what I would say, the neuropharmacology of the brain, meaning the neurotransmitter systems and things like that. So from an epilepsy perspective or seizure perspective, that's kind of what I have been studying. And then the newer theories that are being advanced demonstrate that these ketones have signaling properties much like a drug or a hormone. Okay. And part of that signaling can be epigenetic. So the balance of the data would suggest that this ketone supplementation or elevation of ketones achieved with the diet or supplementation or both together would be therapeutic for this disorder. As a basic science researcher, that's why we are doing this research. there are benefits. That would be I don't know, but I think it could be it should definitely nutrition we feel should definitely be part of an overall program for Kabuki syndrome, and nutritional ketosis offers a wide range of benefits for patients that have Kabuki syndrome. So I'm comfortable in saying that at this point.
Do you think that she's asking if you think the ketones will be available outside of the U.S. When it moves into that phase, specifically in Canada where she's located?
Yeah, I know a number of people are already supplying exogenous ketone supplements to Canada, and I mean, I think there's Amazon in Canada, so you can look on Amazon, and some of the trusted brand names and sources of ketone supplements are on my ketonutrition.org website, where we have the ones listed there are things that we've actually like tested, you know, on ourselves and measured our blood with. So we can provide a link to the website and the different resources there. But I believe which results that are being used in the Angelman syndrome trial should be available in Canada too.
Okay. And then her last question was, what is the expected timeline for ketones to come to market? So, and I don't know.
They're already on the market, yeah. But there's ketone salts, and then there's ketone esters too. So a ketone ester is a more potent form of an exogenous ketone, and the taste is something that needs to be worked on. And also, because they are more potent, there's a potential for side effects and toxicity. So these things need to be sort of evaluated more extensively. But there are many forces at work to get these into clinical trials. So they may be marketed or available as a medical food, and they may be available, you know, as to the general public, but that has not sort of been determined yet.
Yeah, BHB salts are the only ones.
Ketone salts or beta hydroxybutyrate salts are available now, and mixing the salts with medium chain triglyceride oil or powdered forms of the oil are sort of what we are studying now, and they are available.
So we are using regularly available supplements in our trial.
Do you know if the salts are available in Europe? Is that just off the top of your head? Are you?
So I know that a lot of them are on Amazon. So I'm pretty sure certain products on Amazon are shipped to multiple different countries.
And different countries have different regulatory processes. So
Sure.
I communicate with people in Europe that I know have been able to acquire the ketone salts, but I have not been sort of up on all the regulations for each of the countries. But I know many companies will ship to Europe.
Mm-hmm. Okay, great, thank you. She's watching, so
yeah.
Yeah. Well, Corinne just asked if she said you guys are doing ketones and MCT in the mice, correct? Whereas in the human trials, it's just BHB. Is that accurate?
Well, yeah. So because MCT oil can be problematic for some people, and I think the company is going in the direction to actually do another group that would be BHB and MCT. But at the doses needed, I think there was a little bit of a concern with the MCT oil sort of causing some GI distress. But I think there was a discussion of using the two combined together. But that would be introducing two different variables. So I think the first thing was to just see if ketone salts alone could have a therapeutic effect, and then start testing other formulas. But the first thing was to just test the ketone salt by itself, just pure beta hydroxybutyrate, because that was the molecule of interest, and just doing it with a pure beta-hydroxybutyrate supplement made the most sense to do that, at least for the first, round of people.
Well, thank you. So this will be the last question because our time is up. I know you have traveling. I do want to say too, real quickly, to everybody watching, there were several questions that came in that were clinically related that we will ask outside, maybe with our medical advisors, not something related to this live. So we did get your questions. We're not ignoring them. We'll handle them separately. So the last question for you, Dom, would be, how do the results from the studies of mice relate back to our kids? And did you design the testing to reflect the human disease process?
Yes, so the mice have the same mutation, right? So they have been somewhat characterized by Bjornson. They have similar facial structures as the human model of Kabuki syndrome. They have the same genetic mutation, and they have a lower methylation rate on their histone site, just like the human disorder. So that is what is known. We, in our study, have designed to compare our tissue analysis with a blood analysis. So we are going to be looking at the histone methylation rates in a few tissues and then compare it to the histone methylation rates in blood to hopefully increase the translatability into a clinical trial. So then, they just have to take a blood sample from the patient and they can know if it's working, if they need a higher dose or what's going on, because it gets a lot more complicated when you have to take tissues from a human. So we're...
And the behavioral, like right up front, to look at things like anxiety, cognitive function, muscle strength too with the grip test. So we're doing a variety of tests to really understand from a pure behavioral perspective the potential therapeutic value that the therapy will have, in addition to, and correlating that with the molecular studies that we are collecting blood and tissue for. So in that way, I think we're using a mouse model that is, the gold standard. There's not too many mouse models out there, but it is as far as mouse models go, this is a very good model in that, Kabuki syndrome is relatively kind of simple because it's a very specific genetic defect, and having a mouse model with that specific defect is very valuable for evaluating potential therapeutic strategies. So that's a big plus of this study, and I think the behavioral studies and molecular studies were carefully chosen to have the maximum impact, we feel, as it relates to translatability from the preclinical to the clinical study.
Excellent. Well, thank you very much. I will keep an eye on any additional questions that come in and send you guys an email. And for everybody watching, we will share this on ATK's page, on our Facebook page. And by next week, we'll get it on the website. Dom will send some information to you, make sure everything's accurate so we can update your tab. We do have some information on Dr. Dominic's links and stuff on our website under the research section. So we'll get that updated. We appreciate your time, and we will connect again down the road to give everybody an update, and whatever our community can do to help, we would absolutely like to be a part of that. So thank you very much.
Thank you, Renee.
Thank you both. You guys have a wonderful weekend, and we'll talk soon.
Thank you, you too.
Thank you.
Bye-bye.
Bye, guys.
Bye.